All right, so here I'm pressing the record. And
There's a reptile show. There's a reptile show this weekend. Pembroke Pines.
let's see.
You want to come with me, my friend? She'll probably drive Saturday. Anytime.
This will work.
Until I think it ends at 4, yeah. Lame. Busy,
You talking to me?
busy.
Okay, so let's catch up from where we stopped last time. And I don't understand why this thing is not allowing me to use my little tool. Let me see one sec. Okay, so now hopefully that will work. I have too many windows open here.
A week, and it's just like whatever, you just get used to it. Because clearly this is the same thing as you and the guy. You know what I'm saying?
What?
Sorry.
Oh,
Thank you.
all right. Okay. Well, here we go for where we left off with some important concepts, and then let's see how much we can get into the epithelial tissue and into tissues at all. All right, so basically when, oh, am I not sharing?
No, you are not sharing.
Oh, great. I thought I was sharing. A second.
An hour.
So let's share this. What is going on here?
And also,
All right, so now it is sharing.
I'm leaving you. Because you're not kidding.
All right, so Ray, please mute your mic because it's getting some interference here. And then if anyone has a question, please unmute yourself and ask. You there?
Yeah, you just told me to mute it.
Yeah, it's here. All right, cool. All right, so here we go. When we are looking at the inside of a cell, right? So we see that you have that semi-permeable barrier. So here we have our semi-permeable barrier. And when you look at the inside of the cell, we're going to see that we find a lot of stuff there. Okay, so you're going to have proteins. These are the big green dots here. You're going to have ions like potassium, sodium, and chloride. But mainly inside of the cell, you're going to have a predominance of potassium ions and proteins. And then proteins, a lot of times, of course, they have negative charge. So overall, it is going to have some negative charge inside of the cell more than what you have on the outside of the cell. So what happens here? How is it that you get this charge separation, which is going to give you this electrochemical gradient? Okay, so what happens here? How is it that you get a charge separation? Let's see, inside of the cell, if you have the same amount of ions or the same charge of ions than you have to the outside on the outside of the cell, like if you have 10 positive charges inside, 10 positive charges outside, and so on, then you don't have any potential to this membrane. You don't have a difference in how much charge is accumulated in each side. So no potential means that nothing is alive over there. Everything is dead. Okay. So now let's say that you start pumping more positive charges to the outside than you pump to the inside of the cell. So the more positive charges that you're pumping outside are like 3 sodium ions. So 3 sodium ions get pumped out, 2 potassium ions get pumped in. And yeah, sodium and potassium, they do have the same charge, but if you pump more positive charge out, then you pump positive charges in. Now you're going to start having an accumulation of positive charges outside. And we didn't even start dealing with the negative charges here, okay? But just by accumulating positive charges outside, you're going to have a deficit of positive charge in the inside. So that means you are going to have charge separation. It's not like you have negative charge inside, but you have less positive charge inside, which means negative charge inside. Okay. So now when you look at this accumulation of negative charges inside, because you have less positive charge, what happens is that you are going to see that your positive charges on the outside are going to be attracting in general those negative charges that are on the inside, because of course opposite charges attract each other. Okay, so your positive charges are going to be indirectly here interacting and attracting your negative charges. So the inside of your membrane is going to be relatively negative, and your outside of the membrane is going to be positive. So this is your electrochemical gradient. And how do you get that electrochemical gradient? Like you can measure watching, this video over here. How you get there is actually by the sodium-potassium pump. Right? You pump 3 sodiums out and 2 potassiums in. And if you can just measure across that membrane on the inside, you're going to see that it is relatively negative. Like depending on the cell, not all the cells have the same resting potential, but in average, you're going to have negative 70 millivolts right on the inside of the cell close to the membrane. So here, how do you get the separation of charge? The most important anti-port molecule that you have is the sodium-potassium pump. Okay, so this is your sodium-potassium pump. And it is active transport because it uses ATP. And remember that you are using this ATP to change the conformation of this channel. So this is a transmembrane integral protein transmembrane channel, right? So when it binds to ATP, it forms a conformation to dump this 3 sodiums out. When you hydrolyze ATP out of there, it forms a conformation to dump the 2 potassiums in. And this dumping of ions is done against their concentration gradient, meaning that you already have more sodium outside of the cell than you have on the inside of the cell, okay? And you're still dumping more, so that is against your concentration gradient, right? So this type of transport is called primary active transport. Make sure you make a note of that. This is a very basic and important concept. Am I going to ask you about that? Nods, like up and down nods, yes. Okay, I can see in my little video here if the screen is showing my face or not. Is it? Do you see me there or no?
Yes, we can see you. And yes, they are nodding.
Oh, okay, good. All right. So that's the time when I go, clue. Yes, I'm going to ask about that. And the other thing that is very interesting to ask about is about this indirect form of transport, meaning a secondary active transport. So this guy, primary active, directly uses ATP. This other guy here, it's not using ATP, but it depends on a concentration gradient of sodium. So you have to have more sodium outside of the cell in order for this channel here, which is a symporter, okay, in order for this channel here to do its work. So what is it that this channel is doing? This channel is doing symport, so both things go to one side, right? In this case, from the outside to the inside of the cell. Both things are going to one side, and it is carrying glucose with sodium. So this is a sodium glucose symport channel. Okay, what does it do? Well, let's see what's going to do here. If the cell is going to absorb glucose from the interstitial fluid or from the blood, okay, it is going to push the glucose against this concentration gradient because inside of the cell, supposedly you have more glucose molecules than you have on the outside of the cell. So this channel, if you just open the channel and allow osmosis, allow diffusion, not osmosis, you allow diffusion to happen, what is going to happen to the glucose? Well, it's going to come out of the cell because you have more in the inside than you have on the outside. So you don't want that to happen. So what's going to happen here is that in order for you to push glucose against this concentration gradient, this channel works as a syn port using the electronegative gradient or the concentration gradient that sodium has on the outside of the cell. You have a lot more sodium outside than you have on the inside. So this guy is going to go down its concentration gradient. Now, when it goes down its concentration gradient, it will carry at the same time this molecule of glucose, and both of them will go inside of the cell. So sodium is going to go down its concentration gradient, but at the same time, glucose is going up its concentration gradient. Okay? So make a note of that. This is a syn porter. This is the anti-porter. Okay? That's primary active transport. The other one is secondary active transport. Why active? Because it depended on the primary active transport for this concentration gradient to form. Okay. So now get ready because we're going to go to the first iClicker question. And here it is. Okay. When movement of sodium ions down their concentration gradient drives the transport of other substances like glucose across the cell membrane, it is called what? So let's start the iClicker, please.
All right, starting.
And give the correct answer.
30 seconds.
Okay, so we got 30 seconds. So I wanted to start the countdown with the timer, but oh well. Next
Okay,
one.
Doctor, was there nothing showing up for some people? But I have like 60 responses, so I don't know. You need to
Is
buy
it on
it.
the, or is it on the
Okay, so a lot of people have problems. We have 76 responses, but a lot of people are saying like their free trial expired. Some people bought the full membership, but they're still not seeing it.
That's a problem.
Yeah, I
Let's
can't
do the
really fix
best
that.
we can with what we have. Is it counting down or counting up?
It was counting down.
So 30 seconds counting down?
Well, it's over now, but yeah, it was 30 seconds.
Mm.
We had 72 responses. Is someone who is working? Did you guys buy it and like now you bought it or you bought it like weeks ago? Can you restart the app? That's my only idea.
What about the remotes? Did the remotes work?
Yeah, how are the remotes doing? Remotes are doing fine. Yay. Okay, so can I restart the question to see if people can get it?
All right,
I mean,
let's
if you want the extra credit, you'll
let's
have to buy it.
restart. But if you already got your points, don't double dip, okay?
Okay, so I'm gonna start. How's it
All right,
looking?
let's see if everybody can get their
Got
points
it?
in.
Okay, so restart your apps, guys. If it's open and it's not working, but you bought it, delete it from your history and open it again, because apparently that will work. Okay, 15 seconds.
Oh, now I don't know if they have enough
If
time
you
to
haven't
restart.
bought it yet, guys, it's just one extra credit thing. It's like it's not going to ruin your whole grade, so don't worry too much about it, but I would buy it as soon as you can. Yeah. Yes.
Okay, so you got more answers?
We have 49 plus 72. So that's
Basically
maybe
everybody in the class. I don't know how many people in the class, but let's hope that everybody's getting their extra points. If you're going to the class, you might as well get extra credit, right?
yeah.
Okay, so what's your answer?
B.
B, that's your secondary active transport. We just went
I
over it
forgot
and
to
it's
select the
pretty easy
correct
right
answer for
now.
the other one too.
So right, it's really, really easy right now, but during the exam time, it might not be as obvious.
Look at that, I can go back.
So here, now when we look at vesicular, okay, when you look at vesicular transport, sorry, okay. what is it that you're talking about? Well, you're not talking about ions or little molecules. You're basically talking about large stuff. Okay. So you're talking about big, bulky molecules and also fluids, because sometimes you can just like the cell gulps a whole bunch of fluid that is in the interstitial fluid, and whatever is in that fluid gets to go to the inside of the cell. Okay. So gulping stuff, not little tiny stuff, but big stuff, and bringing it into the cell, that is a method of vesicular transport. On the other hand, if you have stuff inside of the cell and you want to dump it outside of the cell, that's also vesicular transport. So here we have a case of endocytosis. When you talk about, endocytosis is gobbling up stuff like macromolecules. When you talk about gobbling up liquids, you call that pinocytosis. Okay. So when you dump stuff outside of the cell, that is your exocytosis. Now, there are some cases in which you pick up something on the outside of the cell, you bring it through the cytoplasm and you dump it outside of the cell. So that's called transcytosis. You just went through the cell and got dumped on the other side. That type of mechanism is seen in the intestinal lining when you're transporting antibody molecules across the cell to be dumped in the lumen of the intestines. Okay, so that's a cold transport process too. So when you look here at endocytosis, here you see this is the cell cytoplasm. This is the outside of the cell. So here you have this formation of a little cup in the membrane. And as you see, this process is using a lot of proteins that are inside of the cell. So this protein, in order for it to be aggregating and coordinated, this protein, this type of mechanism uses energy. All right, so endocytosis, that is using this little coat, actually the name of the protein is clathrin. So this is a clathrin coat of this vesicle. This uses up energy. Okay, so this is a form of endocytosis. Now, another form of endocytosis is here phagocytosis, right, in which this cell here, which is a white blood cell, which is called a phagocyte, this white blood cell is a cell of your innate immune system and is actually eating a very unhappy bacterium over there. Oh, it looks like a bacterium, kind of. So it is this blob of stuff over there. Okay, so this cell here, this phagocyte, gets close to this other cell. It recognizes that it might be harmful, so it will eat up this other cell. And it's amazing that this other cell is almost the same size. Usually the cells, like if it is a little a bacterium, is like a fraction of that size of the phagocyte. But this one might actually be a cancer cell or a malignant cell or something, some dead cell is almost the same size as this cell that is eating it. So this process is called eating up stuff, phagocytosis, cell that eats up stuff. So here we see also that exocytosis, right, dumping things that are in the vesicle here to the outside of the cell. And here comes another question, so get ready. So I will read you the question first, and then you start the timer, Ray. Okay, so everybody has a chance to get in their answers. Okay, so here, in areas of the body exposed to external environment and pathogens, such as in the lungs, okay, which type of vesicular transport would you expect to be the most prevalent? So now you have to see what is the function of the area. You have to defend yourself against foreign stuff. So what kind of vesicular transport are you expecting to see? All right, Ray, let's start.
Okay, I'm starting now, and 30 seconds. $15 seconds. 10 seconds. 5. All right, what's the correct answer? C.
It is C. All right, we just talked about this phagocytosis. So now we're going to take a deeper look in the cytoplasm. Okay. Well, just to make sure that we are in the right page. So next Wednesday, a week exactly from today, is our test, right? Everybody's aware of that? Yes?
Yes, everybody's aware.
All right. Make sure that you bring to class blue Scantron, bring a pencil, bring a razor, okay? Because it's going to be in the class. Unfortunately, I will not be able to be there in person, but Ray and 5 other proctors are going to be looking around. I will be hopefully at the airport and I will try to be on Zoom because my flight is going to be like 9 o'clock, but I have to be there early. So anyway, I will try to be on Zoom and just make sure to, be looking over your shoulders. Okay. So make sure that you're prepared for Wednesday to take your first exam in class, in person. Any questions about the exam? Well, you should be asking what's going to be on the test. Well, all the material that you cover until Monday, including Monday, that's going to be on the test. All right? That's fair game. Okay. I don't know if we're going to get to
On the
the,
self-study, right?
sorry?
Dr. Wizard, including all the self-study?
Yes, yes. If it is self-study, if I put that up, you should know it. So make sure that, you ask questions, you clarify any doubts that you have, because whatever I say, if that was self-study, if that was you're supposed to cover, you're supposed to know it. Okay? All right, so here, getting to the cytoplasm, deeper in the cytoplasm, what is it that you have in the cytoplasm? Well, you have a bunch of stuff. You have the cytosol, and the cytosol is actually, look at this, a gel-like solution. Is actually, it sounds like easy water, right? It's a gel-like stuff made up of water. Isn't that interesting? The book gives you the description of easy water, but it doesn't explicitly say it's easy water, right? But it is. It is easy water. So you have this gel-like state of water, and you have some macromolecules and molecules and proteins and salts and stuff that is dissolved and interacting with that state of water. And so this is the soup of the cytosol. And then you have these inclusion bodies, okay? So you have, what do you have in there? You have some molecules that are soluble, and then you have some molecules that are inside certain membrane enclosed spaces, okay? So you have these glycogen granules, you have some pigments, you have lipids that go inside, also inside of these granules, and you have some pockets with gases, so those are vacuoles, okay? And you have also some little crystals, hopefully not many of those, right? And then you have those organelles. Those are very, very important for the metabolism of the cell. So they're used to build things up, break things down, and each organelle has its specialized function. All right, so we look in the cytosol, what you're going to see. Well, first thing you should keep in mind, you have more potassium inside of the cell, meaning in the cytosol, than you have in the outside. You have a lot more proteins inside of the cell, right, than you have on the outside. And then you have everything else that the cell uses. You have carbohydrates and fats and the storage of things and everything and droplets. Then everything's going to be inside of the cytosol. So here we see that if you look at cells, for example, that have granules, for example, cells that are storing fat, you see these huge blobs. So that's a granule. Okay, you see this huge blob. And actually, these granules, these fat granules, are taking most of the cell space. If you look at the side here, you see the little nucleus are squished to the side. And then these granules take up most of the cytosol if this cell is a fat storage cell. Okay. So besides that, you have the cytoskeleton, that thing that keeps the cell together so the cell doesn't just burst. Okay. And by having a cytoskeleton, when you compare it with bacterial or prokaryotic cells, you see that you don't have the need for this outside cell wall if you have the cytoskeleton, right? So the cytoskeleton will keep a certain shape for the cell function. So what is it that's going to make up the cytoskeleton? You're going to have several types of filaments. You have microfilaments. Those are made up of actin protein. Okay, and then you have intermediate filaments, and you have thick filaments, right? Besides all those for the cytoskeleton, you also have microtubules. So here is a picture of the cell cytoskeleton showing the green, the bluish color showing the nucleus here. So now what do you see? You see actin microfilaments. So this is your actin network, okay? And what else you see here? Basically, you see all microfilaments and stress fibers. You don't see other types of filaments here. So when you start looking at the organelles, you're going to have two types of organelles. You have the ones that are surrounded by membranes, and then you have the ones that are not surrounded by membranes. So we looked here at organelles, like cytoskeleton also falls into the category of organelles. So cytoskeleton and centrioles, of course, your microtubules come from the centrioles, right? So cytokelantins and centrioles, as well as ribosomes. So those are your free ribosomes, as well as the ribosomes that are attached to the membrane. So all of these don't have membranes around them. So those are non-membranous organelles. And then you have the other ones that do have the membranes. And those are mitochondria, ER, Golgi, and lysosome and peroxisome. And I'm going to see briefly the function of each one of those. So you have to know already at the surface what is it that those are doing. Because of course this is not cell biology class, so this is more of a review of organelle function. Okay. So here we look at the mitochondria that, of course, has not only one membrane, it has two sets of membranes, right? It has two membranes here. So basically, when you look at the mitochondria, you see that is very responsible for formation of ATP, okay? So it undergoes respiration, and you can get a formation of ATP by the electrochemical gradient across the mitochondrial membrane, okay? But this is not the only thing the mitochondria is doing. Actually, I have seen before that the mitochondria is actually underwent a symbiotic process, because it used to be bacterium before, right? So the cell engulfed the mitochondria, and the mitochondria became very comfortable inside of the cell, and it started to help the cell providing energy or ATP, and it also got helped by the cell that would provide everything else that mitochondria would need, okay, like sugars and fats, for example, right? So besides that, when you look at the mitochondria, you see that it's not just an organelle, it's like it's an independent organelle in a sense that it will replicate on its own. It has its own DNA, okay, so it has own mitochondrial DNA that is circular. Comparison to other prokaryotes is also circular, right? And it will fuse, like join to two more mitochondria, or it will divide by fission. So it undergoes fission and fusion depending on the metabolic demands of the cell. So here's another picture showing the mitochondria. And here is electron micrograph showing the crystal of the mitochondria. And it is really at this crystal where you have ATP formation for cellular respiration. Okay. Following, we see here a non-membranous organelle. So this is your ribosome. So the ribosome here is showing a large and a small subunit, I believe. Can't quite tell them apart. Basically, the ribosome is responsible for a synthesis of proteins. Okay, so ribosome, you have free floating or you have attached to organelles. So the free ribosomes, they have a different function than the ribosomes that are attached to the ER, right? So free ribosomes, they're going to produce proteins that are going to stay in the cytoplasm. So those are your soluble proteins. When you look at the ER-bound ribosomes or membrane-bound ribosomes, those are going to produce different types of proteins. They're going to either produce proteins for export, so proteins for exocytosis out of the cell, or they are going to be producing proteins that are going to be incorporated into some membranes, right? So those are the ones that are attached to the ER. And here comes the ER. Actually, notice here, the ER is continuous with the outer membrane of the nucleus. It's kind of an extension of that outer membrane, right? So ER that is associated with the ribosome, that is your rough ER. And as you go away from this site close to the nucleus, then this rough ER is not rough anymore. It becomes smooth. It doesn't have these little dots of ribosomes on top of it anymore. So when it becomes smooth, you call it smooth ER. Okay. Now the rough ER is of course associated with synthesis of those proteins that are targeted for exocytosis or membrane components. But the rough ER does something else. It is important for production of metabolic proteins, okay, and also for the production of lipids. All right, so lipids and metabolic proteins are produced in the ER, okay, metabolism of lipids, okay, produced in the ER. Besides, it also is important for detoxification, and depending on the cells that you are looking at, like for example in the skeletal muscle cells or also cardiac muscle cells, the ER is a very important storage or site for storage of calcium. Okay, so this is all part of your smooth ER. Again, keep in mind that the inside of the cell is usually negatively charged. Okay, so here I have a little description on your smooth ER type of work. Okay. And then we get to the Golgi. So the Golgi is right after your smooth ER. So your smooth ER will send out little blobs of vesicles that are going to be incorporated into this Golgi vesicular network here. Okay, so things that are being delivered from the ER to the Golgi, those are destined to be secreted or they're destined to be incorporated in some membrane complex. Okay, so when you look at the movement of things from the ER, you're going to find that some vesicles are going to dump stuff outside of the cell, and some other vesicles are just going to transport things into membrane-bound organelles as well. Okay, so you have secretory vesicles, you have some other vesicles that are going to transport this thing to the ER. And besides that, you also have vesicles that are going to bring things from the cytoplasm back to the Golgi. Okay, so you have vesicles that go to the outside, vesicles that go to the inside, and you have vesicles that go to membrane-bound organelles. Okay, so let me see here. Cells that store large quantities of chemicals to be released do so in structures called what? Oh, this is a very easy one. How do things get dumped to the outside of the cell?
Time has started. Fifteen seconds. Five seconds. All right, what's the correct answer? D.
D, all right, vesicles. Okay, so let's look at some more membrane enclosed vesicles. So here we have your peroxisomes. So these guys are very important for detoxification. Okay, they have some enzymes that are very powerful at breaking up things that could be harmful to the cell. Okay, so they not only neutralize toxins, but they can also be breaking down harmful substances and such things as like free radicals. They take care of neutralizing free radicals. Here's another one. This is the lysosome. It's also very important for cellular digestion. So the lysosomes have cellular enzymes. Okay, they're very good at breaking down like little pieces that got phagocytosed, like for example, bacteria, right? So they chop up the bacteria. They have these digestive enzymes. They can also do some oxidation. Okay. Let me see here the recycling part of lysosomes. Okay, if some organelle is getting close to the turnover state or needs to be recycled, then this organelle is brought to the lysosome, and the lysosome will digest it and recycle the building blocks of those organelles. Okay, so you're coming to more points today. That's the next slide. Oh, you got like so many points today. Okay, which organelle contains enzymes that detoxifies harmful substances? So detoxification.
All right, I'm starting it now. Fifteen seconds. Five seconds. All right, what's the correct answer? C.
Yeah, peroxisomes. All right. So this is the type of questions that you can expect to find in the exam. Okay, so generalized questions on functions of these organelles. Oops. Okay. So here, when you look at all these things that are bound by membrane inside of the cell, so you call that your endomembrane system. So what is the stuff that we just talked about? You have the ER. So of course that is all the ER and then the Golgi and vesicles, lysosomes, everything here that is enclosed by a membrane, including the nucleus and the plasma membrane itself, right? So this is your endomembrane system. Why is this system important? Well, basically you have compartmentalization of cellular function. Okay. So here is showing you again fibers or proteins that are important for your cytoskeleton. So we have three types of filaments that are important for the cytoskeleton. You have the thin one, the intermediate one, and the thick one. So thin one is made up of actin. You have intermediate one that is made up of some subunits that I forgot exactly what proteins those are, but they are intermediate size. They are not just this coiled structure that you find with actin. And then you have very thick ones that tend to be hollow. Those are your microtubules, and they are made up of tubulin proteins. All right, so now finally we get to chapter 4, and we're going to start talking about the tissues that make up the body. All right, so this is the living fabric. So here we're going to first see how is it that you can see these tissues under the microscope, and then you're going to find out about the 4 different types of tissues. And then look at this, 4 different types of tissues. So everything in your body is going to fall under one of these categories of tissues. Okay, so you're going to have epithelial tissue, then connective tissue, then muscle tissue, and then nervous tissue. So everything that is making all your body stuff. This is going to be made up of one of these tissues. Okay. Okay, so now let's see here. Okay. So when you look at individual cells, you're looking at what is it that the cell is expressing and what is the function of the cells. Now, when you put several cells that have the same function, when you put them together, same functional cells, same type of cells, then you get the tissues. Okay, so tissues is just a bunch of cells that are similar in structure and function that you put them all together. Okay, now let me see here. Okay, when you're looking at what is the function of the tissues, you're really looking at histology. Okay, so histology is studying the function of tissues. Now, keep in mind that if you put two or more tissues together, then you get an organ. Okay, remember the classification of small to largest type of organization, right? So cells get together, form the tissues, tissues get together and form the organs. So how do you see tissues under a microscope? Because if you get a bunch of cells and put it under a microscope, you're not going to see much. So first, you have to make sure your cell is fixed. Because if you put a cell that is not fixed or preserved, then the cell might just break or die and you're not going to see anything. Okay, so you have to preserve the cell by fixing it. Okay, then you have to make sure that your certain slide or slice of tissue is very thin. Otherwise, light will not go through and not going to be able to see anything. And lastly, you have to stain the samples so you can actually distinguish what is it that you are looking at. So there are several forms of staining processes that will use different colors that can allow you to see what's in the tissue. Okay. So here we're getting to the epithelial tissue. What is it you call the epithelium? So if it is covering something, it's epithelial tissue. Okay, like covering what? Well, you can have covering from like at the outside of the body, or you can have covering in the inside of the body. So you can cover outside, and you can cover here. You can do covering of the inside that you call lining of the cavities. So everything here is called epithelial when it's doing some covering, okay? So covering outside, of course, is your skin. Now, if you look at the inside surface of your guts, for example, it is also epithelial tissue. Okay, so it's lining epithelial tissue. Besides that, you also have another type of epithelial tissue that's the glandular type of epithelium. Those make up the glands that will secrete substances to the outside of the body. Okay, or yeah, basically to the outside of the body. So an example here is giving us salivary glands. Okay, so glandular epithelium or covering and lining epithelium, two types of epithelium tissues. So if you look at what are the functions, mainly is protection. Okay, besides protection, like of course for your skin is protection, but besides protection, if you see the tissues that are covering the lining, right, of your gut, you don't want only to have protection, but you also want to allow absorption. So protection and absorption. And then you look at the tissue that is covering your kidneys, for example, you also want to have another level, another functional level, which is filtration, right? So very obvious, like intuitive functions for your epithelial tissue. You have your protection, okay? You have absorption, filtration, and of course, if you want to dump things out of the body, you have excretion. So now let's start looking at the characteristics of the cells that are making up the epithelial tissue. So they're very interesting cells. Look at this. First thing that you notice here is that they have a certain polarity, okay? The polarity means that they are different at different sites. They are different depending on what side of the cell you're looking at, okay? So when you look here at polarity, you're going to say that the one side of the cell has one characteristic, the other side of the cell has a different characteristic. I don't know why my pointer is just not working. Sorry, I'm trying to get this pointer here to work. Okay, if the pen works, fine, I'll work with the pen. All right, so polarity. Another characteristic of these cells is that they have contact with each other. They want to attach to each other because, as you imagine, if this is epithelial tissue that is supposed to be protecting you from outside environment, if the cells don't make a barrier, then you cannot protect yourself from the outside environment, right? So they have different surfaces from top and bottom. They have a top and a bottom, and they are attached to each other. Another very, very important characteristic is that they do not have blood vessels. It's very, very important. No blood vessels. That means avascular. Okay. So that is important because you don't want to allow harmful substances or pathogens to invade the body. Okay. So if this is covering all the surface, you really want to keep those pathogens outside. But then that is a problem because if this epithelial tissue does not have blood vessels, how is it that they get nutrients, right? Basically, they depend on diffusion to get their nutrients. They depend on diffusion from the connective tissue that is close to them. Okay, so they have to get the nutrients that is coming from the tissue underneath. Okay, so no blood vessels, but they do have nerves. All right, so that if something goes wrong with your skin, or if something happens to the epithelial tissue, you should be able to feel it, right? So they do have nerves. Another important property is that they can fix themselves in case of damage. So they do have this regenerative property. Okay. And another characteristic is that right under the connective tissue, you are going to find supporting material. So under the epithelial tissue, you're going to have supporting material. Basically, you're going to have your connective tissue that is going to hold this epithelial tissue in place, right? So here, what does it mean to be polar? Well, basically have a top and a bottom. The top part is usually the thing that is in contact with the outside. And then the bottom part is where you're going to find a lot of the organelles, and it's going to be closest to the connective tissue underneath. Okay. So what is this connective tissue that is underneath your epithelium? Well, you have a special name for it. The connective tissue here is called basement membrane. Okay, and the basement membrane will have, okay, I have this thing is going on by itself. Okay, so your basement, ignore that. The basement membrane basically has a couple of parts. It will have a part that is very close to the epithelial. Oh my, I don't know where this thing came from. A part that is close to the epithelium that is called the basal lamina, and it will have a part that is closest to the connective tissue, and that is called the reticular lamina. Okay, pay attention to the red here. Don't pay attention to the highlighter. I don't know why it's going to the to that thing. I don't know why it's going on by itself. I probably saved it without deleting the movements from last class. Okay, so let's look here at this basement membrane and what are the components and why is it important, okay? So first thing here that we see is that you have this basal lamina that is closest to the epithelial tissue, Now, what secretes this basal lamina is really stuff that is coming from your epithelial cells. Okay, so epithelial cells are going to secrete the proteins that are going to make this thin basal lamina, like right there. Then right after this, you have the other part of the membrane, which is called reticular lamina. So what is going to secrete the proteins from your reticular lamina? That is going to be your connective tissue that is right under that reticular lamina. Okay. So again, here, if you look at your epithelial cell, you'll see that you have different sides. So here we have your polarity. The part of the cell that's in contact with the outside, that is your apical surface. Okay. So this is the part that is not connecting to your basement side. Apical surface on the top. And then if you look at the bottom of those cells, that's going to be here your basal surface. If you look between those cells, you're going to have your lateral surfaces. The lateral surfaces are going to connect the cells, the epithelial cells to each other. Again, here we see that you have this very important avascularity aspect of your epithelium. Okay, so here, when you look between those cells that are forming the epithelium, you don't see blood vessels. Okay, you look here, you don't see blood vessels, and you're going to see blood vessels down here. You see here connective tissue. After your basement membrane, you're going to have here connective tissue that will have your blood vessels, right? But then you look here at your epithelial tissue, and you're going to see these yellow looking things, and those are the nerves. Okay, so nerves, yes, no blood vessels all the way down there. How is it that your epithelium gets food? Is by diffusion from the connective tissue that is underneath. Okay, so here you get your next question, which actually I just answered it for you. So which portion of an epithelial cell in the stomach would food be in contact? So this is asking you, the lumen of the stomach is in contact with what part of your epithelial cell? Remember what your epithelial cell looks like, the polarity of your epithelial cell.
Okay, I'm starting it. Twenty seconds. 10 seconds. 5 seconds. All right, what's the correct answer? B.
Yeah, that's your apical surface. Okay, so now looking at more characteristics of those epithelial tissues, as I mentioned, they have to be stuck together. The cells have to be stuck together, like stitched together, okay? Because you don't want things to go through those barriers. So this stitching of the cells together, you call those tight junctions, and they're really tight. They're proteins that will lock these cells together, and that will not allow major things to go through these cells. But they do allow very small ions and a little bit of water, but not much. Okay, so stitching them together, those are called your tight junctions. And still you have some other cells that they have even bigger type of stitching and holding on to each other in which you call desmosomes. So these are made up of proteins that have like a button on one side, a button on the other side, and then both buttons are stitched together to really form these strong plaques that will hold the sides of the cells together, so the surface is not going to break. Okay, so tight junctions and desmosomes. And this is actually important. You can imagine in organs such as in your heart, because when your heart is pumping and is going through this whole force, you don't want, or of filling up and pumping, you don't want the cells to come apart. So this is important for keeping the structure of those tissues together. Again, here, this is another slide showing you that the bottom side of your epithelial tissue is connected through this basement membrane. Okay, remember the two parts of the membrane. You have the basal lamina close to the basal side of your cell, and then you have your reticular lamina that is close to the connective tissue. Okay, so you have here basal membrane, and let's see here. This is just a repeat. So here the other important property of epithelial tissue, that if you damage it, then it can regenerate. So when it regenerates, it will re-establish the homeostasis. So regeneration is very important for the tissue, for epithelial tissue. So here, functions of these epithelial tissues. As I mentioned, you have protection. Besides that, you also have absorption. Besides absorption, you also have certain tissues that have certain areas of specialization. Okay? And in this case, you can imagine, for example, epithelial tissues that are on your tongue. Okay, so you have certain specialized cells that allow you to feel taste of things, right? So you have specialization for certain receptors that will send chemical signals, which will be transformed into electrical signals that will get to your brain and tell you that you have certain thing in your mouth. Okay, so a taste of certain thing. Same thing goes with the olfactory system, right? When you smell certain molecules, the molecules will hit the epithelial surface on your nose, on the sensory part, and will interpret the molecular signal and transform that into electrical signal, and then tell your brain that you have smelled something, which is actually fascinating if you stop to think about it. Like a molecule will float around, come to your nose, will land on these specialized cells in the epithelia, and then you recognize the smell of something, but then the molecule never got into your brain. You got electrical signals that came from the specialized tissue. I mean, I think that's fascinating. I never actually stopped to think about it, but yeah, you only feel electrical signals in the brain. Cool stuff. All right. So specialization. And then you have some of those epithelial tissues that are used in secretion, and we'll see the case of glands. All right, so type of cells that you find in epithelial tissues. In epithelia that is not part of glands, okay, non-glandular epithelial tissue, what you're gonna find? You basically have 6 types of arrangements. So you can have cells that are lined up in one file, right? So one cell line close to each other, or you can have cells that are packed on top of each other, okay? So what kind of cells you have besides a single layer or multiple layers, right? Well, basically, the first type of arrangement that you have a single layer of cells, you call that simple. So that's simple. Now, when you have more than one layer, meaning 2 or more, then you don't call that complicated. You call that stratified. Okay, 2 or more layers, that's all stratified. Only one layer, that's all simple. Okay, so now let's see what are the cells that can come up on those layers, or as a single file, single line. Okay, if the cell is flat, you call that squamous. If the cell looks like a cube, you call that cuboidal. If the cell looks like a column, you call that columnar. All right, that's simple enough. Either it's squamous, that is flat, or cube, or columnar. So you can have what kind of arrangement here? You can have simple squamous, simple cuboidal, or simple columnar. On the other hand, if you have cells on top of each other, you can have stratified squamous, stratified cuboidal, stratified columnar. All right, so that's simple enough. I mean, that's stratified actually. Okay. All right, so let's see here a better representation of how you can identify each type of those cells. Yeah. Because when you look at them, sometimes a cuboidal cell could be like squamous, and then you are in doubt, like, is this cuboidal, is this squamous? Well, the best thing to do is really to look at the nucleus of the cell. Okay. If you look at the nucleus of the cell and the nucleus looks like the yolk of a fried egg, like flattened, like yolk of fried egg, you'll see that this is really a squamous cell. So flat type of nucleus is kind of mirroring the overall shape of the cell. Now, if you look at this other cell here, that is a cuboidal cell, and you see this nucleus is this nice little sphere. Okay, so it's not pushed to any side. It's nice little ball here. So that is a telltale sign that this is a cuboidal cell. On the other hand, if you look at columnar cell, you'll see that the nucleus is kind of stretched. Okay, so kind of long, tall nucleus will go with the tall cell. Okay. So here with the classification of the layers, right? Again, if you have one single line of cells, they're of course, together sideways, but not on top of each other, just sideways. So that is your simple layer, right? If you get cells on top of each other, then this is your stratified layer. So now you should be asking, oh, but look at this. You look at the top of this stratified layer, and you find these squamous cells. They look like fried eggs, so those are squamous cells. And this is the apical layer, right? Get in contact with the outside. And then you look deeper into this stratified layer, and you look over here and you say, oh, these nuclei, they're kind of very round in comparison to those that are flat. So these guys here, these guys over here, they look like little cubes, so that's cuboidal. But these guys are close to the basal layer. So how do you classify this entire stratified piece of tissue? How do you classify that? Is it squamous stratified or is it cuboidal stratified? Let's see votes. How many people think it is squamous stratified? Hands in the class. One person. Okay, who is awake?
There's more than one. I'd say a quarter, maybe half the class.
Okay, who thinks this is cuboidal stratified?
Less than that number. Like,
Okay, good, because your answer is, what is it?
wait, people are confused of like if you're talking about the top one or the bottom one.
Oh, that's my whole point. How do you classify this whole thing? Because it's stratified, it's on top of each other. Do you call it squamous or cuboidal?
Squamous, they are saying.
Huh?
They're saying squamous
Squamous. Why?
because it's the
Because
top
you
layer.
have to take in consideration the apical surface, right? The surface that is in contact with the outside here, not with the basement membrane. So you look at the apical surface and it's going to be squamous. So the whole thing, it doesn't matter if you have all the different types in this whole top of each other situation. The whole thing gets classified as squamous, stratified. Okay? Take in consideration the apical side. Classify your stratified layer. All right, so here we're going to see functions here of simple epithelia, that is simple squamous. Okay, so what is it that these cells are doing? What are their uses? How is it they're useful? So basically, you're going to have a function in absorption and secretion and also filtration. Okay, so simple squamous epithelia, you're going to find in places where you have to have easy access for absorption of things. You're going to find that diffusion is going to be very important. for those cells. So you're going to find that surrounding the alveoli in your lungs. You're going to find that in capillaries of blood vessels, okay? And you're also going to find that in the kidneys where you need a lot of filtration to happen, all right? So these cells, again, they look like fried eggs. That's why I put those little eggs there. And one second, this thing is going faster than I intended. All right. And where you're going to find this type of setting, as we mentioned, you're going to have it in body cavities, or you're going to have also on the epithelial or endothelial layer of blood vessels. Okay, here, endothelial layer of blood vessels can also lining the heart, but very thin layer, and it is squamous, and it is simple. So here you're going to have a picture showing the inside of a blood vessel, a section of a blood vessel. So you see here that this capillary is actually is a vein, is not the artery. Okay, so this is a little vein, is a venule, very thin venule. You'll see that the surrounding this endothelium here, this you find one single layer of cells like holding hands, nucleus, very thin, holding hands with another cell. So this is an example of this simple squamous epithelial cells, right? One very thin layer of cells. Another example is when you look at this membrane that is covering the organs. So you call that the mesothelium, okay? So when you look at this little membrane, you take out from this that is covering the organ, you're gonna see also they have only one layer of cells, and this is a protective, right, layer. And you're gonna have these simple squamous epithelial cells. You see they're flat, and although you don't see how flat they are in that case, but they're very, very flat. So that will bring us to the next one, in which you find simple cuboidal epithelial cells, right? So that's very, very important for absorption and secretion functions. Again, how is it that the cells are going to look? They're going to look like little cubes, and the nucleus is going to be very, very round, okay? If you look at cells that are working in your secretory pancreas, you're going to find that you have these tubules that are layered with a one layer of cuboidal cells, and that is important for production of enzymes that are important in digestion. Okay, so this is your secretory pancreas type of cell. It's not your endocrine pancreas, it's a secretory pancreas. Okay, so cuboidal, simple cuboidal epithelial cells. Okay, so let me see another example here. Yeah, looking at the layer of cells, you should be able to identify that this is simple cuboidal. Okay, and that's the main thing they're looking for. Is it flat? No. Is it tall? No. Is it one layer? Yeah, so that's simple. Does the nucleus look like it's a little ball? Yeah, so you can say it is simple cuboidal. So now let's look here at your simple columnar epithelial cells. So those are very cool cells. They're very tall, and they're very much specialized. And on the apical surface, you're going to find a lot of little indentations that they're called microvilli. And that's important to increase the surface area of the apical side of the cell. Okay, and then some of those columnar cells, they even have some hair that will protrude to the lumen or from the apical side. Okay, so they have those microvilli, and a lot of them have this cilia. Okay, and even some of those layers, remember this is simple columnar, they are going to have specialized cells that will help with the production of mucus, for example. Okay, so if you look at the cells in the intestine here, you'll see that it's very important to be producing the mucus. There is another barrier, okay, from yourself to the outside world. But at the same time, you don't want this barrier to be a very strong barrier, because the job of your small intestine is really to absorb nutrients. So you need to have one layer of cells, and you have to have a certain level of protection, but you cannot have stratified cells here. Otherwise, you'll not be able to absorb the nutrients. It would be too much of layers to go through for proper absorption. Okay. Okay, so where do you find the cells? You're going to find it in the digestive tract, also in the gallbladder, and okay, uterine tubes. So those are your simple columnar epithelial cells. We're going to find more about those cells later. Okay. So now let's get to examples of stratified epithelial cells. So the first one that you have here is cells on the skin. Okay, so on the surface of your skin, you're going to have stratified epithelial cells. But then look again here at the top, at the apical layer, you're going to have, you see here those flat cells. So that is squamous, stratified squamous epithelial cell. Again, if you look at the bottom and say, oh, those guys look like they are cubes, but no, you only take the top part of it. Okay. So where do you find this type of on top of each other squamous cells? You're basically going to find it in surfaces that need a lot of protection because they are exposed to a lot of the outside environment, and you don't want to be absorbing things from this outside environment. Of course, the first example is your skin. Okay, so you need a certain level of cells that are packed very tightly and will block absorption of things from the environment. So when you look at the skin, like we're going to see later, you'll see that these cells, the top cells, are dead because they're far away from the source of nutrients that is under here. Remember, no blood vessels there. So as the cells get packed and pile up on top of each other, they get pushed to the apical surface. So the cells that actually get to the apical surface, those don't have any more nutrients. So those cells are dead. Okay. So important for high protection for areas that you have a lot of wear and tear. Mm. So here we're going to find another example of arrangement of these cells. In this case, you find in the esophagus. Okay, so this is the main example that I give, the esophagus. You could think about sweat glands and mammary glands, but the esophagus is really the prototype here of this type of arrangement. So you're going to find your cuboidal, okay? Cuboidal is stratified cuboidal epithelial cell. And this is actually a bit of a puzzle because I mentioned the cuboidal cell is also important for absorption or secretion, but the esophagus is not really doing a lot of secretion. Yeah, it has a mucus layer for protection, right? And definitely not doing absorption in the esophagus. So it's just used for passage of food, but then this passage of food has to be moist, otherwise it's going to get stuck in there, right? But basically, when you look at the cells that are surrounding here, the esophagus, you find these two layers of cuboidal cells. So since it's more than one layer, so this is your stratified cuboidal epithelial cell setting. Okay, so here is the last example that I'm going to give today. And yeah, so next class, I'm going to start with an iClicker question. So make sure you show up for class at the right time to get your points right from the beginning. Okay. So here, the last example is your stratified columnar epithelial cells. And where are you going to find that? Well, in the pharynx, urethra, anus. Okay. So this is the type of cells in which at the top you're going to have the columnar cell at the apical surface, okay, but then under it you're going to have you can have other types of cells. You can have mainly cuboidal cells or other type of supporting cells. Okay. All right, so that's the main thing here. So I'm going to stop it here. And I'm going to hang around for some more time. If you have questions or you want to come up and comment on something, I always hang around for some time.